Hello BIO100. In this video, I'm going to go over some explanation and clarification of that 3.2 quiz that we did for the lecture portion. And the reason why I'm inserting this video where I am is that a lot of what we'll ask you to do for the lab activity this week is very similar to what you did for this particular quiz. So I want to make sure that you understand the quiz before we ask you to do the same thing for the lab activity. Let's see if my slide will let me advance. Hold on. Okay, so this is what was one of the first prompts for quiz 3.2. It asks you to grab a piece of paper and draw and label a test tube series showing dilutions from the homogenate of 10 to the 1 all the way to 10 to the 6. And it says to make sure you're including volumes moved between tubes so that the total volumes for each dilution amount to 10 mils total. So here's an example of what that might look like. This is not something that I did, actually. It's from a previous BIO100 student. So we have our undiluted tube here on the left. We're given the information that the concentration there is 1 times 10 to the 8th CFU per gram. And then we're doing our different dilution tubes here. So this first one, 10 to the 1 here, we'd be adding 1 mil into 9 mils of PBS. Again, we want our total dilution volume to be 10 mils. So in order for each of these to be a 10-fold dilution, what we need to do is add 1 mil from the previous tube to the next one. That's in addition to 9 mils of PBS that's already present. That gives us a total volume of 10 mils. And so if we continue that for the rest of this dilution series, we have 10 to the 1, 10 to the 2, to the 3, to the 4, to the 5, and then, sorry, my head's in the way, but this one down here says 10 to the 6th. And you can also see in this drawing, the student indicated that with each dilution you're doing, it starts to get a little bit more clear, right? Because it's more dilute. So if you start with a really concentrated dirt sample, by the time you get to that 10 to the 6th dilution, it'll be pretty dilute and pretty clear looking. And this, again, over here, this row is our dilution factor. Okay, so for the next part, it says, imagine that the homogenate collected contained a bacterial load of 1 times 10 to the 8th CFU per gram. Return to your drawing and indicate the concentration of bacteria in each of the 10-fold dilution tubes. So here's our drawing again, and then here at the bottom is where we have that concentration listed. So remember, each of these dilution tubes is a factor of 10 dilution compared to the previous one. So if we know that originally we started with 1 times 10 to the 8th CFU per gram, this 10 to the 1 dilution is going to be a factor of 10 lower than that. So it'll be 1 times 10 to the 7th CFU per gram. Make sure when you're indicating the concentration of bacteria, you're including these units here. It's really important to make sure you're including the units of CFU per gram. And then again, if we walk through each of these dilution tubes, the 10 to the 2nd dilution tube is going to be a factor of 10 smaller than the previous one. So this one contains 1 times 10 to the 6th CFU per gram, and so on and so forth, until eventually, again, apologies for my giant head being in the way, this last one here, this 10 to the 6th dilution, will be 1 times 10 to the 2nd CFU per gram. So each one of these tubes, you're getting smaller in your concentration by a factor of 10. Okay, next we are asked to imagine that 0.1 mils from each 10-fold dilution tube is spread onto an agar plate and incubated overnight. So now go back to your drawing, add plates underneath each dilution tube, and then provide the associated colony forming units or CFUs. Now our drawing would look something like this. So at each step, we're adding additional information, right? So this concentration is what we just did last time. Now we are told we're plating 0.1 mils, so that's indicated here for each one. And then we have our agar plate underneath. Since we are plating 0.1 mils, essentially the number of CFUs is going to be another tenfold dilution compared to that concentration we had in the dilution tube. So for the 1 × 10⁷ CFU per gram dilution tube, that was the concentration we had. If we're plating 0.1 mils, that means we're going to see 1 × 10⁶ CFUs on that plate. Again, it's a factor of 10 dilution compared to the concentration. If we were plating a full 1 mil onto the agar plate, then the CFUs listed here would be 1 × 10⁷. But because we're explicitly told we're only plating 0.1 mils, that means our CFUs are going to be a factor of 10 less than this concentration. And again, just like before with the concentration, it's really important to make sure you're putting your units here. Here we're talking about colony forming units that are visible on the plate. And again, my head's in the way, but when we're thinking about which plate is the one that's countable, remember that's between the range of 30 and 300 CFUs. So out of all these dilutions that we did, this plate right here is the only one that would be considered countable. It has 100 CFUs, or 1 × 10⁵. So this is the one that you should have circled that falls in that significantly countable range. And one more thing to mention about this plate in the significantly countable range, we need to identify this one because this is the plate we then want to use to do our back calculations to figure out how much, or confirm how much was actually in that original tube, what the original concentration was. And the reason for that is we are specifically looking for that countable plate because if there are too many colonies there, too numerous to count or too few to count, that means we don't really trust that number of CFUs. So we don't want to use that for calculations because it might not be accurate. So we want to first identify that countable plate and then use the information from that countable plate to do our back calculations. And that was what you were asked to do in question 2 here, which was that multiple choice question. So you were asked to calculate CFU per gram to determine the concentration of bacteria in the cleat homogenate. And you're given this different information here in the table, the dilution factors, the volume that was plated. Note this is given to you in microliters, and the colony forming units. So first of all, remember we want to identify which plate is countable. So looking at these CFUs here, which again is this right-hand column, which of these plates is the countable one? Right, it's this one right here, the 10⁴ dilution factor. It has 158 colony forming units, which falls within that range of between 30 and 300. So this is the plate we'll want to use to do our back calculations. And when we do that, I can move my head. Okay, when we do that, we have the formula here is what we want to use. So CFU per volume plated, and remember that volume plated when you're using it in this equation needs to be in milliliters. So you're identifying the CFUs, dividing by the volume plated in milliliters, and then you're multiplying that number by the dilution factor in order to determine that original CFU per gram concentration of bacteria. So when we do that with this particular question, we have 158 colony forming units. Remember, I said this was given to us in microliters, so we need to convert that to milliliters for use in this particular equation. 100 microliters is equivalent to 0.1 mils. So we get that number, and then multiply that by the dilution factor, which is 10⁴. And so what you end up with is 1.6 × 10⁷ CFU per gram. For this particular quiz, that wasn't an option in multiple choice, so that's why the answer was none of the above. So this is in general how you approach these types of dilution experiments and questions. So hopefully this gives you a little bit better understanding that you can then apply to the lab activity for this week.